In the final moments before a cell divides, two polar hubs orchestrate a violent, highly synchronized event. They cast out a dense web of microtubules to pull the cell's genetic material apart. These hubs are the centrosomes. As the primary microtubule organizing centers in animal cells, they dictate spatial organization, intracellular transport, and overall cell polarity. If you look at an organelle like the nucleus or a mitochondrion, you see a distinct lipid membrane defining its border. The centrosome has no membrane. It is completely exposed to the cytoplasm. This presents a structural problem. Without a physical boundary to contain its components, the centrosome still manages to replicate with the same strict regulation as DNA. A healthy cell mandates that the centrosome duplicates exactly once per cell cycle, no more, no less. To understand how this works, we have to look past the old textbook drawings of a fuzzy organelle and examine the precise, self-assembling molecular geometry at its core. For decades, electron microscopes showed the outer layer of the centrosome as an amorphous, disorganized cloud of protein. Modern super-resolution microscopy presents a very different picture. These microscopy panels reveal distinct annular structures, proving the centrosome possesses a highly ordered nanoscale architecture. At the absolute center lie the centrioles. These are two barrel-shaped microtubule structures oriented at strict right angles to one another. One is the older, fully mature mother centriole, and the other is the younger daughter. Surrounding this core is the pericentriolar material, or PCM. This is the active scaffold where new microtubules are actually born. The structure of the centrosome is entirely dictated by the cell cycle. During the first gap phase, or G1, it rests as a single pair. Its ability to organize the cell depends entirely on how it physically matures through the subsequent phases of the cycle. Looking at this G1 state, the cell contains exactly one resident centrosome, composed of that single engaged mother and daughter centriole pair. As the cell prepares to replicate its DNA in S phase, the centrosome begins its own duplication sequence. The process starts with a biochemical trigger. An enzyme called PLK4 kinase concentrates in a specific ring around the proximal end of the mother centriole. PLK4 phosphorylates a protein called STIL. This chemical modification creates a highly specific binding environment for the next critical building block. That building block is Cas6. As seen in this cartwheel cross-section, Cas6 proteins self-oligomerize, snapping together to form a central hub with 9 radiating spokes, terminating in pinhead structures. This Cas6 cartwheel establishes a rigid 9-fold symmetry. It acts as the internal scaffold required to stabilize the formation of a brand new procentriole. Microtubule triplets, labeled A, B, and C, assemble directly onto the cartwheel's pinheads, elongating at a strict perpendicular angle to the mother. The physical geometry of the Cas6 assembly acts as a biological failsafe. It ensures that exactly one new procentriole forms per mother centriole, preventing runaway duplication. Moving into the G2 phase, the core replication is complete. The objective now shifts to massive organelle maturation. To handle the mechanical stress of impending cell division, the centrosome rapidly expands its footprint by recruiting more pericentriolar material. Far from being a disorganized cloud, the schematic reveals the newly recruited PCM forms a highly ordered, interlinked lattice made of coil-coil proteins. The foundation of this network is pericentrin. It acts as the primary structural strut, anchoring the entire molecular lattice to the outer wall of the centriole. Embedded securely within this pericentrin web are the active payloads, gamma-tubulin ring complexes, or gamma-2 arcs. The gamma-2 arc provides a literal starting template. It locks onto the minus ends of free-floating tubulin dimers, stacking them into a hollow tube. This prevents spontaneous, chaotic microtubule growth out in the cytoplasm. Centrosome maturation is the controlled construction of a massive molecular launchpad, engineered to organize the forces required to split a cell in two. The cycle culminates as the cell enters meiosis. At this stage, the two newly duplicated, fully mature centrosomes separate and deliberately migrate to exact opposite poles of the cell. Operating as spindle poles, they project dense, overlapping arrays of microtubules to physically capture, align, and separate the chromosomes. The accurate division of the cell's entire genome relies entirely on the structural integrity of the centrosomes assembled during the preceding hours. But if a cell exits the division cycle to differentiate into a specialized tissue, the centrosome takes on a completely different role. In this G0 resting state, the centrosome stops organizing the interior of the cell. It physically migrates away from the nucleus and heads outward toward the plasma membrane. As shown in this top right illustration, the older mother centriole utilizes specialized structures called distal and subdistal appendages to physically dock itself into the inner surface of the cell membrane. Once locked in place, the mother centriole acts as a basal body, seating the outward growth of an axoneme to form a rigid projection. The cell's internal division engine is repurposed into a primary cilium. This sensory antenna detects extracellular signals and fluid flow, allowing the cell to communicate with its environment. This multi-state complexity has given rise to the field of centrosomics, the comprehensive mapping of the organelle's dynamic protein network across all its configurations. It operates as a scaffolding cartwheel, a microtubule hub, a mitotic spindle pole, and a sensory antenna, all built without a surrounding membrane. Because it dictates many processes, the physiological stakes are high. If strict duplication licensing fails, the cell ends up with amplified extracentrosomes. This geometric error leads to abnormal cell division, driving the genomic instability characteristic of many cancers. Alternatively, if the centriole fails to dock properly in G0, the resulting loss of cilia causes severe developmental disorders known as ciliopathies. The centrosome is the ultimate cellular architect. Understanding human health requires decoding the precise self-assembling geometry that operates at the